Thanks for joining today. A couple of months ago, I discussed single cell gene expression analysis in the training. And so you can find the recording in LIMS. Um, so today's training will also be recorded and archived in LIMS. You can view these recordings by going to the Sales tab in LIMS and then clicking on Training, and you should see them listed here with links to the video. If you do have trouble finding it, just reach out and I can help you. So for today, I'm going to skip a lot of the basics on 10x genomics chemistry and jump right into the various services that I plan to discuss. I do have a couple introduction slides that I'll go over, but then I'll be going directly into the immune profiling kit to talk about TCR and BCR. Then I'll discuss Cite, Seq, and cell hashing. These are two methods that are very similar. They use antibodies to get the most out of single cell data. And then lastly, if there's time, I'll briefly go over Cellplex, which was just released last week, and it's a 10x genomics product that offers an alternative protocol for cell hashing. So to review a few things, single cell experiments are used when you have heterogeneous samples and you're interested in learning more about the individual cells or the subpopulations of cells in your sample. The platform does require a single cell suspension. So PBMCs are the easiest population to study, and that's what we see in most of our projects. But the platform does also work from other tissues that have been dissociated into single cell suspensions or even single nuclei suspensions. The chemistry is based on capturing cells in emulsions along with barcoded beads. For RNA assays, which is mostly what we do, the RNA gets reverse transcribed. The cDNA then gets a unique cell barcode and UMI added to the sequence and gets amplified and converted to a library. And I listed just a few. I think this is everything we offer from 10x here. 3' gene expression, immune profiling, which includes 5' TCR and BCR, Cite-seq, cell hashing, multiome, and linked read whole genomes. And that's a discontinued kit, but I think we still have a few kits in-house. And so I have this file here, just, why are we talking about single cell? 10x genomics, you can see every year more and more papers get published that reference single cell data, specifically from 10x genomics. They are the forefront of single cell kits platforms. We also offer a Takara single cell kit, but I'm not going to be covering that in today's training. So I'll start off by talking about immune profiling. A large portion of our clients work in the areas related to immunology. So the use of single cell sequencing is critical for the deep understanding of the immune system. 10x genomics can be used to deconvolute immune system heterogeneity. It allows identification of rare and distinct immune cell subsets, which can be used to build development trajectories for immune cells and more. In the adaptive immune system, the combinatorial possibilities lead to such diverse and immense repertoire of T cells and B cells and their antigen specificity. So understanding the VDJ recombination in T cells or somatic hypermutation and class switching in B cells is critical in dissecting cellular interaction in the tumor microenvironment or in the disease response. The value that single cell analysis brings to understanding the adaptive immune system is the ability to resolve both the gene expression profiles within each cell, but also match those gene expression profiles and cell subtype information to corresponding alpha and beta chains in T cells or heavy and light chains in B cells. So as I said, a lot of our clients work in immunology related fields and might benefit from single cell TCR or BCR. So here are just a few examples of ways our clients use this service. The range of applications that our clients use is pretty wide, starting from immuno-oncology. This could be gaining an understanding of tumor microenvironment or tumor mutation burden. infectious disease, we don't process infectious samples, but a lot of clients are able to make their own gems. And if they have a project that studies infectious disease, they're free to make their own gems and send those to us for downstream processing. Vaccine development, that's another big one right now. Uh, Studying autoimmunity, um, immune cell atlasing, or just basic T and B cell repertoire discovery. I also added a note here that for the TCR and BCR kits, they are exclusive to human and mouse as far as making the TCR and BCR libraries. The 5' gene expression part of the kit could be used for any organism, but the immune repertoire profiling is specific to human and mouse right now. So getting into the kit offered by 10x, it's an immune profiling kit. Uh, as I said, it allows for a whole transcriptome, single cell 5' gene expression that allows you to understand immune cell discovery and phenotypic analysis. In addition, the solution also offers enrichment for paired full-length human or mouse VDJ sequences from single T and B cells. The gene expression and VDJ sequences can then be matched in a single cell to simultaneously characterize TCR transcripts, BCR immunoglobulin transcripts, and profile gene expression from any sample. The throughput of the system allows for the profiling of hundreds to tens of thousands of cells, which then allows for rare clonotype and cell type population discovery. And finally, revealing the clonality, diversity, and cellular context of the immune repertoire. This could be visualized in Cell Ranger and then further analyzed in Loop Browser. Actually, this is Loop Browser. So the workflow looks like this. You start with your sample input, single cell suspension. You then use immune profiling reagents from the 5' kit and the Chromium controller to create your gems. You then break the gem and amplify cDNA. And it's this amplified cDNA that's used as the input for either of the 3 libraries that the kit makes. And then you can pull the libraries and sequence together. So the kit itself comes with enough reagents to make a single library from a gem, but you can add more libraries at a minor increased cost. The gem creation is the most expensive part of the kit, so that's really the bulk of the price. Adding a TCR or BCR library or both doesn't add that much cost to the project. And again, there are some clients who may not be interested in gene expression. They may only be interested in the TCR data or the BCR data. So the gene expression itself isn't necessary for the project. Here's an example of the Cell Ranger output. This is from a BCR project. Kushal will later go over the advanced analysis available for TCR and BCR, but this is the basic visualization of the data that you can expect to get from a project. In this case, you can see the estimated number of cells captured, the number of cells with productive VJ spanning pairs. Here you can see just what percentage of it is from heavy chain or light chain data, UMIs, a lot of statistics on sequencing, and just the overall prep itself. For annotation, you can see just how the data was aligned, the context being made, stuff like that. So overall, this just provides a visualization of the data. You also get a page that just shows you the top clonotypes in your sample and how frequently those clonotypes appear in the data. So moving along to Cite. So there are many important features that measuring many different phenotypes and okay, sorry. So moving along to Cite. So you can get a lot of information by measuring different phenotypes and taking a multi-omic approach. CiteSeq, for example, takes both gene expression data and cell surface protein data and combines that data together to get a more refined analysis of your cell population. Um, so, for example, your gene expression data might tell you about the cell types in your sample. It might tell you about the subpopulations. But if you don't get enough genes expressed for certain cells, those cells will be considered dropouts and they're not included in your overall data. There's not enough information to uh, to assign those cells to maybe a cluster or to group it together with other cells in the same population. And so adding the cell surface protein data to it just gives you that extra layer of information on the phenotype of your cells. You can see which cell markers are being expressed. This can tell you about the type of cell that it is. It can tell you about the state of the cell, and you know, uh, maybe even tell you more about the lineage of the cell. Um, the so CytSeq uses antibodies in this little cartoon, which target specific cell markers. So some examples I listed here, immune cell markers are CD19, CD3, CD16. These antibodies, which I'll go over in a second, have an oligonucleotide conjugate, which has a barcode attached to it. And from this data, you're learning more about the different cells in your sample. Um, so the key takeaway here is that CiteSeq uses antibodies that target specific cell markers. On the other hand, very similar chemistry is cell hashing. Um, Cell hashing also uses antibodies, but these antibodies target ubiquitous cell surface proteins, something that you would find in all cells, such as CD298 or beta 2 microglobulin. Um, so the point of cell hashing, unlike CiteSeq, uh, it's not to learn more about the cell population itself, but it's to multiplex your samples into a single gem and then be able to deconvolute the data after. So in this cartoon, you can see there are 4 different samples to be processed. Under the normal chemistry, they would be processed into 4 individual gems. But with cell hashing, you could label them with these antibodies, um, hashtag antibody 1, 2, 3, 4. This is the nomenclature that BioLegend uses. And once each each sample is labeled with a different antibody, you can then combine them together, make a single gem, sequence it, and from the data, deconvolute the samples and separate out the four um, the 4 samples. So again, the main difference here is these antibodies are targeting specific cell markers that will identify the cell or the state of the cell. And these antibodies just target ubiquitous cell markers. So for CytSeq and cell hashing, we use BioLegend, TotalSeq A, B, and C in our lab. And there are a few differences between each of them, and it could be a little confusing to tell them apart. And I think a lot of that comes from TotalSeq A, which is sort of the odd man out of the group. Um, TotalSeq A was originally meant to be for cell hashing and CiteSeq combined. And so it's not supported by 10x Genomics. You buy the antibody oligoconjugates from BioLegend, and you can they have a poly A tail on them, so they are used in combination with the 3' gene expression kit. Um, you label your samples with these antibodies, and then you can end up making a hashtag library, a site library, and a gene expression library. Um, So, again, originally this was meant to be a way to do cell hashing using a 10x kit in a way that's not supported by 10x. Um, but since then, TotalSeq B and C have also added hashtag antibodies to their antibody list. And so it kind of gets a little confusing at that point. So I'll try my best to clarify. Um, but TotalSeq B is and C, these are both supported by 10x Genomics. The main difference here is that TotalSeq B uses the 3' gene expression kit with feature barcoding. TotalSeq C uses the 5' gene expression kit with feature barcoding. Um, and I know this seems like, hey, this also uses the 3' kit, but they have different capture sequences. So that's the main difference. They have different PCR handles. Um, TotalSeq B and C can be done completely start to end using um, using antibodies purchased from BioLegend, but then the rest of it can be done using a 10x genomics kit. TotalSeq A requires reagents that you have to purchase independently, just PCR master mixes, primers. Um, and just for the lab end of things, it's important to keep in mind which one you're doing, if it's TotalSeq B, TotalSeq A, just because the capture sequence is different for each, and also the PCR handles are different. I drew them all in red, but um, the TotalSeq A requires an HDO primer for the hashtag library, an ADT primer for the Cite library, and then both TotalSeq B and C just use the cDNA primers plus, I believe it's called, from the feature barcode kit from 10x. So going into cell hashing, just who is this used for? Again, the main point of it is to combine your samples and make a single gem. So of course, the first advantage of cell hashing is that it can increase the throughput of a project. You can process more samples with fewer reagents. So of course, that's tied to a reduced sample cost. Um, by combining your samples together and running them through the same chip and making the same gem, you're also minimizing batch effects. Um, so you can see this as two different ways. You can imagine some clients will be interested in this just for the cost saving. I want to process 10 samples and pay for one gem. So that makes sense. But on the other side, there are other clients who may not have very many cells to make individual gems. We usually ask for at least 100,000 cells per gem. And sometimes the population of interest is so small that a client can only get 10 or 15,000 cells. So in that case, if we use hashtag antibodies to label those smaller cell populations, and then later combine them together. Combining enough samples would get us to that 100,000 cell point where we can then confidently make a gem and sequence the cells. Um, the drawback to cell hashing is that it must be used on viable cells. It cannot be used on methanol fixed cells or nuclei. So ideally, it would be used for cryopreserved cells or a fresh sample drop. And then one other note here. So in this example, this is just a t-SNE plot from gene expression data from a cell hashing project we did. So you can see just from gene expression alone, we get certain clusters, but there's no obvious difference between the four samples that are pulled together. But once you get to the antibody data, if you generate the t-SNE from that, you can see four obvious clusters here representing the four samples pulled together. For our lab, currently we're limiting cell hashing to 4 samples maximum to be pulled together. And the reason for that is because the cell prep just still takes a long time. And to get 4 samples stained with antibodies, counted and pulled together, that's kind of the limits to what one person can do. Technically, they do make more hashtag antibodies, so you could do, I think, up to maybe a dozen for some of these, but it would just require more people in the lab. And of course, then you get into problems where some of the cells or the samples might have QC issues, and we would probably have to communicate with the client to figure out do we proceed with, some of these that pass QC, or, I don't know. It gets complicated the more samples you're processing. So this is the basic workflow for Cite, specifically for total seq B or C. Um, so you start with your single cell suspension, you stain it with antibodies, and after a couple washes, you create your gem. You do your PCR amplification here, but unlike what I showed you earlier with just the TCR BCR sample, you end up with two tubes because you are also amplifying the antibody oligotag. So you do a size selection at this point, and you end up with two tubes, the larger fragment, which is the amplified cDNA, and the smaller fragment, which is the amplified antibody oligotag. Um, so you still end up making, you can still make your gene expression library from the amplified cDNA, and that's true for both total seq B and C, 3' or 5'. If you're doing total seq C, which is 5', you also have the opportunity to make a TCR or BCR library, or both, um, from this amplified cDNA. And then lastly, you make a Cite library from the low molecular weight fraction. Um, and all of these can be sequenced together. And then the data can be used after to study the different types of cells in your sample. I also made this slide. It just shows sort of how complicated things can get with total seq A mixed into the workflow. Um, so again, you start with your single cell suspension stained with antibodies. You create your single gem. If you're doing cell hashing, then this would be a pool of cells that were stained with different hashtag antibodies. You still do your PCR here, but if you're doing total seq A, then you'll end up with two different products in your low molecular weight fraction. You'll end up with both the Cite-seq and the hashtag libraries being amplified here. Um, and then again, based on the workflow you have, you can still have your gene expression TCR, BCR libraries. So it does get complicated at the bottom. I just, this is just for reference later on. I just put a list of the different types of libraries that can possibly be made with each of the three workflows. So total seq A, you can have a gene expression, hashtag, and possibly either a Cite library as well, or sometimes it could just be gene expression and Cite, gene expression and hashtag. Total seq B, you'll have your gene expression library, and you'll have your Cite library. Again, they just started making these hashtag antibodies for total seq B and C, so you could also do hashtagging. But in this case, they'll be part of the same library. The same for total seq C. Total seq A is the only one where you'll end up with two separate libraries here because they have different PCR handles on the antibody oligo. So very quickly, I'll go into multiplets really quick and sort of dive deeper into the benefits of cell hashing, and then I'll pass it over to Kushal. So an important consideration when deciding how many cells to target for 10x is the cell multiplet rate. So I think the main drawback to 10x chemistry is you're loading cells into the chromium, and there's a multiplet rate. The more cells you load into the chromium, the more likely you are to have cells that have multiple cells captured in the same emulsion. So they call that multiplets. You could have 2, 3 cells in the same emulsion. And the problem with that is once the multiple cells will get tagged with the same cell barcode. And when you generate data, it's not very easy to identify those cells and omit them from your research. So most of our projects will aim to capture 5 or 6,000 cells. And so we get a 4 to 4.8% multiplet rate, which is certainly manageable. But you can imagine 10,000 cells, now 8% of your data has multiplets, and you have no idea which they are, which 8% that is. You can go higher, and again, this is a linear increase. If you do 20,000 cells captured, 16% of your data will be multiplets, and so on. So one other benefit of cell hashing is that it provides a way for you to identify these multiplets. So in these four little cartoons down here, you can see when there are no hashtags used at all, some of these emulsions will contain one single cell, some will contain two cells, some will contain three, but there's no way to definitively uh, tell which of these data represents the multiplets and which are the singlets. Once you start cell hashing, and now you have two different uh, samples that were pulled together, labeled with different antibody barcodes, you can then look for cells that have two different antibody barcodes in the same cell, and you'll immediately know that's a multiplet, and you can omit that from your data. So in this case, this cell could be omit. You can omit this one, and also this one. It still does leave a certain fraction of cells that will have multiplets that have the exact, the same antibody barcode, but um, the more cell hashtagging you do, the more different antibody barcodes you add to your sample, the more likely it is that you'll get a mix of two different barcodes, two or more different barcodes in your multiplets. And so you will be able to identify and remove those multiplets from your data. So that's just another advantage of using cell hashing. Uh, Again, the ones that end up having the two cells with the same barcode, two cells with the same antibody barcode and the same emulsion, there's not much you can do about that, but this is a great way to clean out your data from all the other multiplets. And I will end on this slide. This is just to sort of give an overview. Based on my last slide, I said you can now identify a lot of these multiplets in your data. So this cap of 10,000 cells that 10x recommends capturing, um, you know, that that gave you 8% multiplets in your data. Now you can remove a lot of those. So the next obvious step would be, okay, let's capture more cells, 20,000, 30,000, 40,000 cells. So this is just to show that there is a limit um, to this approach. So on this graph, you can see these are just this is the amount of reads you would have to generate if you capture 20,000 cells, um, 25,000, 35, etc. The blue are data that would represent singlets, and based on the percentage of multiplets, this reddish orange color is how much data would be lost to multiplets. And in yellow, I just did a quick calculation of the sequencing cost, not including library prep or anything, just for data generation. And you can see there's diminishing returns after a certain point. The sweet spot seems to be around 30,000 cells captured, where anything beyond that, you're paying a large chunk more. But it's a very few of the reads are now useful for your project. A lot of them are just going to this multiplet rate. Um, Same thing here when you make the jump from 35,000 to 45,000 to 40,000 cells, you're seeing a large increase in cost, but not a large increase in useful data from your samples. So when it comes to what they call super loading, loading more than 10,000 cells into a chip, um, you know, this is something to keep in mind. 30,000 seems to be the max that would be recommended. Um, and even then, the cells themselves are very difficult to count at a concentration so high. So there's still a lot of variability at that point. and a lack of precision for loading, for super loading. But it certainly can be done. And with that, I'm going to go ahead and stop. As for the Cellplex, the new kit from 10x Genomics, I'll add a few slides to this just for reference, but it, it would take a while to go through, and so maybe that could be for a later training. But if there are any questions specifically about this, maybe I should share my screen again in case I need to reference a slide. Feel free to ask questions.
Derek, this is Addy. Just a quick question around Cite and the specific antibodies. So, I would imagine that with clients that we're looking at, they would have very specific antibody needs as well, right? So, when we order these from 10x, is the question that we have to revalidate some of this again, or the fact that we have validated Cite just allows us to get the specific antibody in question and do the experiment for them, right? I'm just trying to get a sense of how replicable it can be or not, right? As we look for Cite customers.
Yeah, totally. That's a great question. So I generally, I think Angelica and Olga also just refer people to the BioLegend website. They have a lot of antibodies, antibody oligoconjugates for sale, including hashtag and for site. And so the drawback there is most of them are immune cell based, but they've all been validated. They all, come at a concentration that's optimal for staining cells. So for the most part, we just steer them there to BioLegend's website. And if it's something very custom, there it's possible to have BioLegend create these antibody oligoconjugates for them, but at that point, that's when you have to start validating and titrating to figure out what is the antibody concentration that should be used for the Cite.
Thanks a lot. That's helpful.
Okay, then I will pass it over to Kushal.
All right, let me share my screen. So I think Derek and I spoke, and it kind of made sense that he go through the lab part of it, and then I can come and talk about the analysis. That would be more seamless, and the flow would be better off there. Okay, does everyone see my slides?
Yep.
Okay.
Yeah.
So I thought I'd first walk you through the Mango report and what's there, right? So typically once the lab finishes sequencing, the data are uploaded to the LIMS, and we initialize a basic workflow sort of analysis. And the 10x processing mainly involves doing a basic data QC, and then 10x's proprietary cell ranger software, which Derek showed a few screenshots of basically how the data would look like, is performed, and then we provide basic reports. And this would be sort of everything that's part of the basic report is up until the single cell RNA advanced pipeline, and it contains basic information about the projects, um, project statistics, and the cell ranger report, which can be downloaded as loop files, which the client can then basically play around with. Not sure why this isn't working, but uh, essentially, once this is done, what we do is, the whole problem with 10x's cell ranger and loop browser is that it has very limited functionality. Once the data are generated and aggregated by cell ranger, then the loop files can be downloaded by clients, and they can play around on their own with some of this data, but there's no real flexibility in advanced analysis beyond some of the clustering that loop provides. So what you're seeing here are t-SNE plots of the cells that are clustered based on the average expression profiles of individual cells. And there's not a lot of flexibility in saying, okay, I know for a fact that there are only 4 clusters, so can you rearrange or reorganize these labels based on those clustering assignments? And really there's not a workaround around that situation. And pretty much this is the only sort of analysis you can do. There are some heat map representations that cell ranger and loop provides, but not a lot of flexibility. Um, so what we do is beyond the basic reports that we provide, which is cell ranger and loop, we do a number of downstream analysis steps, including QC and pre-processing and secondary and tertiary analyses. And so one of the first things that we do is an initial QC. As Derek was mentioning, 10x is inherently challenging the data because there could be cases where you have multiplets, you have doublets, which is probably the most frequent in gene expression data. And because cell viability is a critical factor in maintaining the robustness of the underlying gene expression data, we need to be able to clean out basically cells that are not viable or they're dying. They have a high number of mitochondrial reads, which could sometimes be an artifact of tumor biology, because tumor cells are rapidly dividing. There could be a lot of mitochondrial gene expression. And so what you're seeing here is an initial QC plot that we always take a look at. And we filter cells based on a number of user-defined criteria, such as the number of unique genes detected in each cell, meaning remove all the doublets and triplets and multiplets, remove empty droplets with no barcodes, or remove low-quality cells with fewer reads, or remove cells that have a number of reads mapping to mitochondrial genome, which could indicate low quality or dying cells. And so what you're seeing here is a distribution of the cells. So each dot here is a cell, and the violin plot shows the distribution, number of features, the number of cells, and the percentage of mitochondrial uh, potential mitochondrial reads. And so, this is sort of a manual intervention step here. So the bioinformatician, whoever is analyzing, would take a look at this sort of QC plot for every project that requires advanced analysis, and then decides on cutoffs based on this distribution that we see for these three metrics. At any point, if you have questions, please feel free to stop and ask. Uh, okay, so once we clean out some of the dirty data, so to speak, from the previous step, we then need to normalize the data so that we can actually compare expression profiles across cells, because they can have variable expression between cells, right? So you need to normalize for sequencing depth and overall expression profiles in each cell. And then instead of using the entire set of, let's say, 20,000 genes that are typically expressed or fewer in a cell, what we can do is sort of optimize performance and compute times by identifying the most variable set of genes that can explain the differences between the cells. So that's called feature selection. So once we normalize, we do feature selection to identify the subset of features that can best sort of explain the data that you're looking at. And this set of features are typically used downstream for analyses such as PCA, heat maps, clustering, and so on. So we typically select the top 2,000 most variable features or 3,000 most variable features. And the idea is, can these top 2,000 or 3,000 genes actually explain the majority of variation that you see in your data? And that is used also for principal component analysis, which is a way of reducing the dimensionality of your data, because you have, let's say, 10,000 cells and each cell has 20,000 genes or 15,000 genes that are expressed. You're essentially looking at a matrix of 15,000 cross 10,000. So how can you reduce that amount of complexity into something that can help you identify the sort of the most interesting patterns in your data, right? So that is essentially dimensionality reduction. What you're seeing here are principal component plots, which show that, uh, principal component 1 and principal component 2 can sort of account for more than 30 to 40% of all the variation in your data. And subsequent principal components, which contain probably genes or cells that still contain genes that are not as informative, but if you add more and more components to your data, you can sort of recover most of the information that's contained in your data set. And these would be the genes within the components that sort of explain the most amount of variance. So this is just fraction of variance explained by the data. So now we've sort of determined, let's say, the top 5 principal components explain 60% of the data. But how much more do you need to include to sort of overcome any sort of inherent noise in this data set, right? Because unlike bulk gene expression data, single cell gene expression data is pretty noisy because you're literally trying to capture gene expression from a single cell, and so the noise to back signal ratio is pretty high, and so you want to optimize the number of genes that you can select to truly distinguish noise from signal, right? And so how many components do you choose? 10, 20, 100? And so a quick way to do that is something called an elbow plot, which basically ranks principal components based on the total variance in the data that they can represent. And so it's called an elbow plot because it almost represents an elbow with the rest of the forearm and the wrist. And so what's typically done is the point at which this elbow sort of starts flattening, you can pick that number as the number of components to choose. So in this case, we say, okay, I'm going to use the top 10 principal components to then go and do advanced analyses like clustering, heat maps, and so on. And so with that, we can take the top 10 to 12 principal components. We typically start a little higher because if 10, we start with say 13 or 14, and then we keep coming down and seeing if this clustering changes or does not change basically as you add more principal components. And so this is essentially how we arrive at t-SNE clusters, right? So uh, in order to be able to identify or ascertain how many potential clusters of these cells could be there, we do all of these steps beforehand. So it's not a very straightforward process, and so I think a lot of customers lose track of this sort of amount of work that goes into creating these t-SNE plots. But these t-SNE plots are somewhat meaningless, right? What do these mean? It doesn't. So one question that customers often ask is this distance meaningful? Does this mean these clusters are very different from these? So it's not a one-to-one relationship in that sense. There's another representation called UMAP, which is sort of more meaningful nowadays. And all this says is, these cells are more similar to each other in this cluster than these cells. So the idea of clustering is to minimize the so-called differences within a cluster and maximize the differences between clusters. Once we've identified the clusters, we can then do differential gene expression. And differential gene expression here is not across samples as it is typically done in RNA-seq, but it's across cell types. So if we've identified 8 clusters, we can look at the top genes that can explain the variants or the differences between clusters. So you're seeing cluster specific gene expression profiles that seem to separate them. And it sort of is a validation that the clustering that we've performed is actually correct. But these knee clusters by themselves are sort of meaningless. You don't know what the cell types are, right? So there are two ways to sort of identify cell type markers and assign cluster labels essentially. So, they're typically lysozymes, CD8As, or other immune cell types. Or, if you're not interested in immunology, then you can look at developmental biology. Also, there are examples of single cell clustering to identify different cell types in the pancreas, beta cells, alpha cells, islets, and so on. And so if you have a set of known markers, we can then manually look at the expression profiles of each gene that is known to be expressed in a particular cell type and manually assign those clusters the labels that correspond to the genes and their expression profiles. So for instance, if you are looking at specific immune cell types, then we'd look at the underlying genes for each of these cell types and assign cluster labels based on the expression profiles of the individual genes for that cell type. The other sort of way is to automate identification of this, because sometimes manual curation can be a little bit arbitrary. There are cases where, for instance, CD8 T and NK cells could share a few markers and the expression could be very similar. So how do you really distinguish between these two? And so there are softwares and algorithms that can automate some of these assignments. And that's one tool that we use is called single R. It's widely used in the field, and it annotates cells using a number of different built-in reference databases. what it does is it looks at the average expression profiles of each of the clusters we identify, and it compares that against a known database of gene expression profiles for a number of different cell types, and it assigns it based on if there's concordance between the database and our data set. We also can provide individualized gene expression profiles by cell type. So if the client or customer is interested in a number of different gene markers, we can then look at the expression profiles of the genes across the cell types for each of those genes. And so this shows you, strength of the expression as indicated by the color of the circles, and the size of the circle represents basically what fraction of the cells are these genes expressed in. So the larger the dot, it just means that a particular gene is expressed in predominantly the entire cluster, and the color of the dot indicates the overall expression level. So that's sort of the gist of the single cell gene expression data analysis that we perform. There are a few other things that we can typically look at, but that's more customized. But this would be sort of the standard advanced analysis pipeline that we typically run. Any questions until this part? Okay. The next step that the other thing that we can do is to combine single cell immune profiling data with gene expression data. And what that allows us to do is to correlate gene expression changes with, immune responses in the body or in the cell. And that is particularly interesting in cancer drug discovery, in immunology, and vaccine development. And so that is also sort of rapidly growing in the time that I've been doing this, and I've been seeing. And we recently finished analyzing a data set for a client out of Ohio State, where they were looking at patient responses to a certain drug, um, both in terms of gene expression profiles and the TCR repertoire and the changes in the TCR repertoire space. So we start off TCR or BCR analysis by providing a number of summary statistics. So we look at the overall clonal proportion, meaning what are the top clonotypes that are unique across the samples. So this shows you the overall clonotype space proportion, uh, meaning what fraction of the most unique indices represent the entire clonotype space that is present in a sample. So another way to think about this is, in red, what red here shows is the top 10 clonotypes. So the top 10 clonotypes actually occupy maybe, 10% of the total space, the first 10 clonotype indices basically. The next 100 clonotype indices represent the next 15%. So there are about 7,000 clonotypes that represent about 40%, I would say, of the entire clonotype sequence space that you see in the sample. So this sort of is very helpful because if there are one or two clones that are very dominant, then you would see this red bar sort of stretch across this. So in this case, 10%, the top 10 clonotypes, or the 10 clonotypes in this particular sample, that actually occupy almost 20% of the entire repertoire. So this could mean, these TCR sequences are actually maybe immunogenic, and they're actually recognizing some specific stimulus in the cell. There's also rare clonal population analyses that we do. In this case, there's one clonotype that's very predominantly occupying the majority of the clonal repertoire, but there are some clonotypes that are less than 5%. And so this gives you sort of the flip side view of the first plot that I was talking about. We can also look across samples and see if samples share the same TCR CDR3 sequences. That is also very interesting when you're looking at immune responses. Are specific CDR3 sequences or antibodies, recognizing the same antigen, and that is also very useful when you're doing drug discovery studies, looking at immunogenicity of your antibody. And then we provide a number of statistics on the CDR3 sequences itself, which is the most important part of the antibody, right? So we look at the overall frequency and usage of these individual TCR beta variable genes, or the immunoglobulin heavy chain variable genes. We look at the usage of these genes across samples. Are there any preferences for specific heavy chain or light chain variable genes between samples? And again, that can indicate, biology rather than just sequencing that you're looking at, right? We also profile the overall lengths of CDR3s because this also has a significance in the biology and what it could possibly mean in the context of the research question you're asking. And we also look at the frequency of the individual gene segments, and this again helps you identify the most dominant type of variable gene that's being used in your sample. We can also look at a particular clonotype across time. So let's say there are samples that are uh, you're tracking longitudinally across time a sample that is sequenced for its TCR repertoire before a drug treatment and it is sequenced after a drug treatment. You can look at specific clonotypes that are expanding, right? So that is probably the most definitive way of identifying TCR antibodies or BCR antibodies that are responding to a challenge. So let's say pre-vaccine and post-vaccine, are there antibodies that can be um, detected that are actually expanding, which means they're actually responding to the vaccine, right? So that those are the sorts of things that can be very easily identified from such analyses. And what you're seeing here are the top 7 clonotypes and how they are sort of being shared across samples. And in this particular sample, you see this clonotype here, this first guy here, actually is not even present in the sample, but this light blue one here slightly increases, and there's another one that's almost not present in the sample but is hugely expanded in this other sample. So these are the sort of the clonal dynamics that you can observe with these sorts of plots. You can also integrate your single cell gene expression data. So let's say we've identified 10 clusters. How do the TCR or BCR antibody sequences uh, correspond to these clusters? Are they shared across clusters? And so what this shows you is a one-to-one relationship between the TCR sequences present in cluster C2, for instance, and that some of the antibody sequences here are also seen in another cluster C6. So this is a chord diagram that shows that relationship. We also do diversity index score calculation, and TCR diversity is just a measure of how uh, abundant your TCR antibody sequences are and how rich your clonal space is. So think of this, an analogy to this would be, a glass jar filled with, different liquids of different viscosity. And you're just looking at how many different types of liquids you can essentially fill in to a glass jar. And you're comparing the types of liquids and the different viscosities across different jars. So TCR diversity usually corresponds to, obviously underlying biology, and in the context of drug response or vaccines, you ideally want just a fewer sets of TCR sequences that are clonally amplified, because those are the ones that are actually responding to whatever stimulus that's being given. So the lower the diversity, the better it is, essentially, because if you have too many different TCR sequences, then it doesn't mean that there are any particular types that are actually responding to a stimulus. So these are just 4 different types of indices, and each index score actually is a different sort of representation of what you're trying to see in your samples. Are you trying to identify the most dominant TCR sequence? Are you trying to identify rare populations? And so these scores actually correspond to each of those types of questions that the, the client or the customer may have. Another sort of neat thing we can do is overlay some of these TCR sequence data onto our t-SNE plots from gene expression data. So this is one example where we performed t-SNE clustering. And now the question we're asking is, hey, can you tell me, using much TCR expression data, what are the clonotypes that are expanding, that are small, there are only single sequences, or there's no TCR information at all for some of these clusters. Right? And so you see these particular 2 clusters here seem to be concentrated with the TCR antibody sequences. And these are actually CD4 and CD8 positive T cells. So that again sort of validates our clustering, right? Previously, we identified these clusters as T cells, and that is exactly where the antibody sequences are also expressed. And within these, we then ask, okay, so I see this particular cluster here is a large cluster, meaning there's one or two sequences that's hyper expanded. I then take the top TCR sequences and then I ask where are those sequences, in which cells are they? And that's exactly where we see these. So there's one big cluster of TCR clonotypes and then there's another, and that's exactly where the expanding TCR clonotypes are also present. So this is a very useful sort of plot that a lot of our clients really appreciate. And so this is also another thing that we can do. So I've just quickly run through some of the analyses we've done. I hope that was helpful to all of you. And just to summarize, we offer these pipelines to pretty much all single cell gene expression and immune profiling data sets. And our ability to integrate both expression and immune profiling data, I think, is pretty powerful. We use a number of different tools for the analyses and visualization. And so I think that is where our advantage is really. Yeah, so I can stop now and take any questions.
So
Kushal,
very
this is
cool,
Adi.
Kushal. Oh, go ahead.
Sorry, go ahead, Derek.
Oh, you can go first. I'll ask mine after.
So Kushal, I was just wondering, if there was any, I would say, appetite for looking at more of a predictive solution using AI type technology, right? So using like an ensemble platform for AI and trying to predict more of what might happen to go in hand with the more kind of data-driven approaches, right? Is that something that has been explored or you think would be worth exploring?
I'm not sure what you're trying to ask. Predict. What are you? What is your like? What are we trying to predict? Like it depends on
So
the question, right? So
yeah, so typically if you've got that single cell data and you were to load similar type of data into an AI platform, then um, there are some predictive AI models that can help you predict what might happen, right, with specific subpopulations of cells in what you showed with the t-SNE plots. And those are some experiments that have been published in arteriosclerosis and diseases like that,
I
right?
think we're talking about lineage tracing and population dynamics, right? So yeah, we
Right.
could do. It's just that we've not had any request for that, but yeah, I think
Okay.
that's definitely something that can be explored.
Okay. It's something that used to come up where I used to work, but again, I'm not that was much more in a research context, and some of it was used for some of the SARS work that was being undertaken at that time. And not SARS, but SARS-CoV-2, so COVID work. Yeah.
Yeah.
So Kushal, I was just wondering, is there a timeline for having any of these analyses added to Mango? Because I think, especially the TCR, BCR outputs, I think a lot of clients would love those, and
Yeah, the
it would be
TCR
great
is
to.
the TCR is already we have the bulk TCR reports, but I think the next step is to get the single cell TCR analysis also. The single cell gene expression would have to be sort of it has to be divided into two parts because as I showed you initially that QC step is a manual intervention step. So we typically stop the pipeline at that step and then we look at the QC and then we decide the cutoffs and the parameters and then we do the second half of the analysis. But yeah, I'm working with the team to get the TCR and BCR Mango pipeline set up. So hopefully in the next month, within the next month, we should have that.
Yeah, sounds good. Until then, would you be able to share your slides?
Yes, I will. So
I mean, I
all
think you or Sheetal would probably be on most calls and can probably go into details on this, but it would still be good to have just to familiarize myself with it. Cool.
Kushal, what's the format that we would share the data set that's not in Mango?
My plan is to basically have these analyses added as custom analyses tabs in Mango because that was one of the features that was worked into Mango, right? So that you can have your standard pipelines show as, the Mango report, but then any sort of advanced analysis can be added on as new tabs to the Mango reports. That's how I envision it, at least for the advanced single cell. The single cell TCR and BCR, we can just create a separate Mango report. And in any case, it always ends up that we do a slideshow and presentation, which Olga can speak to as well, which we've been doing with our clients. So I don't know if there's a preference to do it within Mango or just save some time and just keep it the way we're doing it right now. I'm okay with either.
So I like the idea of having a custom tab with the advanced analysis plots.
It just makes it consistent and everything is in Mango then.
Yeah, because there are some instances where the commercial team might want to say, hey, these are the standard outputs, but this is the custom outputs that we have done for some of our other clients, but very specific to your case, we can work towards creating such advanced plots. Let us know, right? But the standard output goes to everybody.
Yeah.
Dushal? Sorry, just in terms of positioning this to your clients, how many man hours do you think this will save them as far as analysis goes? Like if they were to do this themselves, how long do you estimate them to take to do this?
Really depends on the number of things that they want to do, right? If it's just a simple gene expression experiment that they want to look at, in terms of heat maps, differential expression, automated clustering and labeling, man hours. I mean, for me, it would take maybe a couple of days. So I don't know. It just depends on their comfort levels with analyzing such data, right? So maybe a week or less, I think.
Yeah, and Olga, if your question was also to estimate how much to charge, also look at it from this perspective. For any advanced analysis, especially for single cell and immune profiling, I think we should look at per sample $300 to $500 as informatics cost, right? If you go by man hours estimate, Kushal is saying 2 to 3 days, it'll be like 20 hours, and you multiply it with per hour rate, it may not justify the value addition that this analysis provides to the particular project. So when possible, try to position a few $100s on a sample basis rather than number of hours spent on it, because number of hours spent includes the subject matter expertise, the iterations that happen internally, which may not be quantifiable specifically, right? So I think look at it from that perspective also when we put the costing for some of these particular advanced analysis.
Sure, absolutely.
Kushal, are these publicly available softwares and tools, or did we have to purchase licenses for any of the?
Publicly available.
Okay.
So, Angelica, a quick question. Do we get questions from customers on what tools are being used for this analysis, or it doesn't matter? What is your experience?
Usually they don't. I mean, sometimes they'll ask, but the reason I was personally asking was for my own understanding on if they have like a team of bioinformaticians in-house, is there some sort of leverage we have there? Like these are softwares that they can't have access to, or they would have to pay therefore like using us. But if it's free, they could easily use it themselves. So I was just that's why I was asking that question personally.
Okay.
And I had some questions for Derek, if you don't mind if I jump back, or I can just ask him off the call since we're over time.
I'm happy to answer. If anybody does have something else they have to do, feel free to go do that, and I'll keep recording, so you can always check back on the recording later to hear the question and answer. Yeah, go for it.
So my first question was for when you're doing hashing, what is the minimum starting cell count per sample that we can work with?
So it's okay. So to answer that question, ideally we still want as many cells as possible. 100,000, 500,000 cells would be great. So that's still to be encouraged, but your question is the minimum that we can process. And I would say maybe 20,000 even would be on the very low end of what we could accept. 20,000 viable cells. The caveat, of course, is that the fewer cells we have, the less precision we'll have when pulling the cells. And so if they give us, 10 or let's say 5 samples that each have 20,000 cells, we can stain them and pull them together. But will we get a 20%, 20, 20, 20, 20 distribution of reads across all 5? Probably not. But we'll get data for all 5. And so if it was a project that couldn't be done with single cell a year ago and now it can be, then that's a win there.
Perfect. And then that kind of answered my other question, but is there a starting viability that you need, or would we do dead cell removal first before staining, or does the staining kind of act as like a double step in that manner?
Yeah, good question. So for this, both for TotalSeq, ABC, the staining doesn't include any sort of like positive selection. You just stain the cells, so you still want high viability cells. Whereas if we were doing something like a CD45 enrichment or a dead cell removal, there's that selection pressure that'll get rid of a lot of the dead cells. But in this case, that's not there. So yeah, we still need, highly viable cells. They recommend 95% viability starting with, but, I think for us, 80% is fine. And, we should be able to get good data from that.
Okay, so if a client had low viability cells, and they were just doing hashing, the first step would be to do dead cell removal if it made sense, and then move forward with the hashing. Is that right?
Yeah, exactly. And so one thing to also keep in mind, and again, it's not a huge deal, but the more antibody staining you'll have to do, which includes the dead cell removal, I think that takes 30 minutes of incubation. If you're doing cell hashing, there's another 30 minutes of incubation. If you're also doing Cite, there's a third 30-minute incubation. So there might be some concern about the transcripts of the cells, the cell states changing from this long period of time of them sitting on ice. Certainly, clumping might be an issue, but there are ways around that. My point is it can be done, but it's not, just keep in mind that like the more hands-on you get with these cells, the less optimal the processing of the cells will be.
Makes sense. And then if someone's doing both hashing and Cite, would the workflow be to hash all the samples, combine them, and then run the Cite, and then tag it with the Cite antibody? Or do you do it?
Yeah, I think
separately?
I think that's the best way to do it. Technically, I'm sure it could be done if you just do a single antibody staining and combine the hashtag antibodies and the Cite antibody pool. But I think I think it could be done. You just have to do some calculations to make sure that the concentrations are still optimal when you do the staining. So yeah, you can probably get away with one staining. I mean, you definitely can, but you could also, break it up into 2 separate 30-minute antibody staining steps.
Perfect. Thanks, Derek. And then Adi, I wanted to expand just a little bit on your question earlier to Derek about validation and in that the question you had about the antibodies. So to my knowledge today, we always have the client purchase their own Cite antibodies since, they'll need to, like, we can help with experimental design, of course, and maybe on case-by-case basis we can help. But since those will be unique to the client's projects, we always just have them buy their own for that. But in terms of the hashing, since those are not unique and those can be used for other client projects, I believe we do stock those. But Derek can clarify on that.
Yeah, we generally have. We generally have a set of 4 total seq A and 4 total seq C hashtag antibodies in stock. That's what we've used for past projects, and so we have excess in each tube. We just haven't had a total seq B project, so we don't have that.
But if we got one, we would buy those, the HTO part ourselves, right?
Yeah, exactly. We would buy the probably 4 of them or whatever, however many samples there are, and then we would keep those for future projects and try to sell that to get rid of, to justify the purchase.
Perfect, thanks. And then, sorry, one last question. I had a client the other day that was under the impression that their library, like they had run a hashing Cite TCR BCR 5' project. So they had 4 libraries per sample, and they were under the impression those had to get sequenced in the same lane. My understanding is that would not matter, but do you have any input on that?
They do not have to be sequenced in the same lane. Yeah, there's just like any other. The data's there. The data's in the library exists. It'll. You can certainly pull all of them together, or sequence them separately, and it would work. Yeah.
Okay, thanks.
Angelica has a very good understanding of the Cite seq and hashtagging projects.
She does, absolutely.
Looks like she has worked in the lab on that.
We've had a few customers back in the day that had to do it, so it was a steep learning hill to get on board with their project.
No, wonderful.
Yeah. They're cool. I like these projects.
Any other questions?
Yeah, Derek, sorry, just to follow up on that hashtagging discussion, you mentioned something about hashing 10 samples in 1 gem. Are we still doing just the 4 samples per gem at maximum?
Yeah, definitely only 4 at this point. Currently, Gavin's the only person with hands in the lab for cell hashing. Technically, Debbie could also jump in as backup, but it just it gets very complicated to have so many samples going through cell prep at the same time because there are so many wash steps. If they're sending cryopreserved cells, viability could become an issue. A dead cell removal, like if you have 10 samples and 2 require dead cell removal, it gets really complicated and hard to track. And even pulling them together, we try to get an equal distribution of cells per sample being pulled, and trying to get 10 samples at an optimal concentration to be pulled together, it gets crazy. So for now, definitely 4s should be the limit. And as we add to our team and more people get trained on cell hashing and they feel more comfortable pulling more samples, I think we can try increasing that.
Okay, sounds good. Thank you.
Great. So I will go ahead and end it here. Yeah, thanks everybody for joining. If you do have any follow-up questions, feel free to send me an email or you can call me because we work at the same company. All right.
Thanks a lot, Derek, Kushal. Thank you very much.
Everyone.
Thanks, Derek. Thanks, Kushal.
Thank you.